This video is on the topic of investigating transpiration. It is possible to investigate the effect of environmental factors on the rate of transpiration using a potometer. Bubble potometers can be used to show the volume of water taken up by measuring the movement of a bubble in a capillary tube, while mass potometers can be weighed to determine the mass of water lost by a plant during transpiration. Information collected using a potometer can be used to calculate the rate of transpiration under different environmental conditions. Setting up a bubble potometer can be quite fiddly, as it is crucial to avoid allowing air bubbles to form anywhere in the system. Air bubbles can form a blockage in the xylem of plant shoots, preventing normal transpiration. Bubbles can be avoided by using a large basin of water during setup. The plant shoot must be cut underwater to prevent air bubbles blocking the xylem vessels. The rest of the equipment must then be assembled underwater to prevent air bubble entry into the potometer itself. An additional sealant, such as Vaseline, can be added underwater too. Once set up on the workbench, the leaves should be dried with paper towel so that water does not block the stomata. Whenever you are describing part of an experimental setup like this, be sure that you can explain the purpose of each of the steps involved. Once the airtight setup is complete, the investigation can begin. We first need to introduce a single bubble into the potometer. We do this by lifting the tube out of the beaker to allow an air bubble to form. The bubble can be positioned by opening the reservoir tap and allowing a small amount of water into the system. At this point, it is a good idea to leave the potometer for a few minutes so that the rate of transpiration can stabilise. We then need to measure how far the bubble travels through the capillary tube in a set amount of time. This can be done by recording the starting position of the air bubble and then using a stopwatch to measure a set period of time, e.g. 20 minutes. At this stage, the position of the bubble can be noted again, and the start and end bubble positions can be used to calculate the distance travelled. The bubble position can then be reset, and the experiment can be repeated in the same environmental conditions. After this, the independent variable can be changed and more repeats carried out. The independent variable is the variable that you choose to change and that is of interest in your investigation. For example, if we are studying the effect of light intensity on transpiration, then light intensity is our independent variable. Light intensity could be changed by altering the distance between the potometer and a lamp. While we change the independent variable, the dependent variable is measured. Here we could choose to measure the distance moved by the bubble in a set time period. Remember that the measurement gained for the dependent variable depends on the change in the independent variable. Now, in order to gain a valid set of results that truly show the effect of the independent variable on the dependent variable, we need to be sure that no other factors will influence the results. These other factors are known as control variables, and they must be kept the same. While investigating the effect of light intensity, for example, you would need to be sure that you are controlling air movement, humidity, and temperature. This exam question asks you to describe the practical method that you would use to investigate the effect of temperature on the rate of transpiration in plants. We know that a detailed method is needed because 6 marks are available. You must include specific details of the method that you will use. For example, you could describe how you will assemble the potometer and the need to repeat your investigation at different temperatures. You must include references to your independent variable, so state how you will control temperature, maybe using a heater with a thermostat, and which temperatures you will include in your experiment. State how you will measure your dependent variable. Here you would need to refer to the distance moved by the bubble and the time period during which the bubble moves. You should also include examples of control variables. These might include the need to control the light intensity and any air movement in the room. When describing practicals in extended questions like this, you should always include details of the method. as well as referring to all of the different variables that are involved. In summary, the effect of environmental factors on transpiration can be investigated using a potometer in a range of environmental conditions. Potometers must be assembled underwater to prevent the entry of air bubbles which might influence the results. The distance moved by an air bubble over a set time period can be used as a measure of the rate of transpiration. It is essential that descriptions of potometer practicals include details of the method, as well as references to independent, dependent, and any control variables.